Good afternoon, everybody. We have finished a lot in the first unit. Now, what is left is biodiversity. So, today we're going to look at biodiversity. Now, if I ask you to write, you have the option to write it with a pen, or you can write it with a pencil. You can write it with a marker. You can use a sketch, right? So you have very different types of things to use to write. So you have diverse ways to write. That means you have many different options. You have very many different varieties to write it. In the same, this is regarding the non-living things. I mean, you can see examples like this in so many different areas and in so many different things. But we, as students of biology, we always relate to living things, right? So when it comes to living organisms, we have already learned in our previous classes that there are single-celled organisms and we have multicellular organisms, right? And not just that, we have different organizational levels, right? From single cell to tissues to organs, organ systems, and ultimately these organ systems make organisms. That means we have diverse variety. There are lots of varieties of species living on this planet Earth. If you take these normal ants that you see going around in all kinds of seasons, especially in summer, you know that there are 20,000 different species of ants out there, although you only see like one or two. I mean, the naked eye, you can say these are red ants, these are black ants. But there are 20,000 different species of ants. This should tell you that there is a vast diversity when it comes to living organisms. This is called biodiversity. Okay? So all the different varieties of the living organisms that inherit in the planet Earth make up the biological diversity or biodiversity. Now, we have already seen, because there are so many millions of organisms, we have classified them, right? There are different ways to go about it, identifying, naming them, classifying them. We've learned that in taxonomy. So it always comes to, in science, we break it down, right, into different parts. So in this biodiversity also, there are different levels of them. So what are the three main levels? Although organizationally, as we have seen, they go up from single cell to multicellular organisms. We are at the we are at the peak of the chain or the pyramid, if you want to look at it that way. But if you take the organizational level, there are three main levels to classify them or to organize them. One is, and the guy who actually popularized popularized this term, biodiversity, as Edward Wilson. What's his name? Name is Edward Wilson. Okay. Remember the scientist's name. So we have seen, we we have three different main levels. One is in the biodiversity, one is the genetic level, the species level, the ecosystem level. Now, if we look at the genetic level, what is this level? The genetic level is within the species. Is within the species. Now the example given is Rauwolfia vomitoria. Victoria. Write it better. Yeah, vomitoria. So we should be small letter, right? Writing the scientific name. This you can see growing in the Himalayan ranges. You see it in the Himalayas. This is one species. What do we get from Rauwolfia vomitoria? We get reserpine. This okay. This is used for this is used this is used in treating high blood pressure. Okay. So we are looking at diversity, biodiversity at the genetic level. That means there is a variation in the species, within the species. And we have taken as an example Rauwolfia vomitoria. This plant, this grows in the Himalayas. And reserpine is what is extracted from this, and it is used for treating high blood pressure. But this plant, it's the same species, but depending on where it is grown, you know the ranges, right? Himalayas are at different altitudes. So the place where you collect matters because the concentration of the reserpine changes. The concentration and the potency. Potency means effectiveness. Concentration is the amount that you get. So these two things, although we're talking about the same plant, these two things differ. There is a diversity, there is a variation at the genetic level within the species when it comes to concentration and the potency of reserpine that is extracted from Rauwolfia vomitoria. Right? So this is at the genetic level. We are talking about biodiversity at different levels. One is genetic, one is species, and the other is the ecosystem level. At the genetic level, we've taken the example of Rauwolfia vomitoria. Depending on where it is grown, the concentration and the potency of it varies. Okay, and the species, when it comes to species diversity, you know that we can see that there's more amphibians in the Western Ghats than the Eastern Ghats. You know the Ghats, right? Yes. Okay. So there's a greater amphibian species in the Western Ghats than in the Eastern Ghats. This is the diversity that we see at the species level. Now, at the ecosystem level, you know what an ecosystem is, right? A self sustaining region within a landmass or water body. But if you take India as an example, we have different kinds of ecosystems. If we start off with from the water, we have coral reefs, we have wetlands, marshes, estuaries, mangroves, we have deserts, and we have mountain ranges. So we have very different ecosystems present in India rather than if we take a Scandinavian country like Norway and Sweden, which is very cold. So the kind of vegetation that you see or the flora and fauna that you see is not as diverse as you see in India. So the biodiversity has three levels. One is genetic level, the other is species level, and the other is the ecosystem level. There are three levels. So the genetic level, it is within the species. The species level, you're talking about different taxa here. There are different wide variety of depending on the habitat. Like let's say if you take Western Ghats, you find more variety of amphibians in the Western Ghats than in the Eastern Ghats. And if you take at the ecosystem level, not all continents or regions have the same kind of ecosystem. We in the tropical regions, we have different climates, and so we have different kinds of ecosystems, starting off from coral reefs to deserts. So these are the three, remember. Now that we have learned about the different levels of biodiversity, there are also indexes or indices. Index is singular, indices is plural. Indices of biodiversity. Now, before we get into the index of it, let's look at the picture. We see, we see here. Okay. So we have seen that there are three different levels, right? So the first is the genetic diversity, which is within the species. If we take the Homo sapiens, we as human beings, one species, right? But we at the um, but we're not all the same, right? There are Africans, there are Indians, there are Mediterraneans, there are Americans, British, Australians. You can see the difference, right off the bat. You can see that they're not from here, from India. So this is the genetic diversity. At the species level, if you look at the water, you can see there are mammals, there are fish, reptiles, and non-chordates also. So this is at the species level. Now at the ecosystem level, there are coral reefs, there are deserts, there are prairies, grasslands, the tundras. These are the different levels of diversity that you see. Three different, right? One is genetic, the other is species, the other one is the ecosystem level. Now getting back to the board, there are indices or index for biodiversity. The first one is alpha. What does alpha mean? There are three. Here also the indexes are three. One is alpha. Let's take a different color. One is alpha, beta, and gamma. So how do you measure the diversity? That's what we have the index for, right? So the alpha kind of measurement is just to count the number of taxa. Or it's usually the species in a particular area. This is the alpha diversity. Now, the beta diversity is if you take two adjacent ecosystems, then you take out the common species between the two and count the unique species. Between the two ecosystems. Usually they are adjacent. Now the gamma index shows the total number of species. In all the ecosystems of a particular area. Now to understand it better, let's look at a picture. This is just a measurement, right? How diverse is the area? If somebody asks you, what's the diversity here? There are three different measurements that you can tell them. One is the alpha, the other is beta, and the third one is the gamma. So let's say this is a site A, this is a site B, and site C. For our understanding, we can take this as three different ecosystems. So the first one, all these different figures that you see inside, they are species, okay, for our understanding as an example. So the site A has 7 different species. 1, 2, 3, 4, 5, 6, 7. So the index, the alpha index, is alpha diversity is 7. The number is 7 because you have 7 different species in this area. Now for B, you can count and there are 5. In the same way for C it is 7. Okay. So that is the alpha diversity. Now coming to the beta diversity, there are for site A, these are two adjacent ecosystems. Sorry, excuse me. So for site, oh my God, what's happening to my throat? So the site A has 7 species and site B has 5 species. We know that the alpha diversity for A is 7 because there are 7 species. The alpha diversity for site B is 5 because we have 5 different species that we have counted. Now, when it comes to beta diversity, what you have to see is what are unique in these two ecosystems. Now, you know that there are crosses in both of them and there are stars in both of them. So you discount these. Unique means one of a kind, right? So you have to count the unique species in both. That means you take these two out because you find both of them in both the ecosystems. So you count the rest of the species. So how many are there? 1, 2, 3, 4, 5, 6, 7, 8. So the beta diversity is 8. Okay. Let's say for site A, the alpha diversity is 7. And beta diversity is, how many did we count? 1, 2, 3, 4, 5, 6, 7, 8. There are 8 unique species. So the beta diversity is 8. Like in the same way you can do for site B and C. Okay. So what are they? You see that there are 1, 2, 3. Same, right? You see 6-arm star and you have a triangle and you have a plus. These 3 are the same in B and C. So if you want to know the beta diversity between site B and site C is 1, 2, 3, 4, 5 and 6 between B and C. But between A and B it is 8. This is beta diversity. Now coming to the gamma diversity, we count all the species. Okay, if we take these two, within, let's say, within this area, there are only two ecosystems, A and B. So you count all the species in A and B. How many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So the gamma diversity will be 10. It looks like the class is something like a math class, huh? A beginner's class. Anyway, so there are different indices, there are different measurements for biodiversity in a particular area. So there are three different indices. What are they? Alpha, beta, and gamma. The alpha diversity means you just count the number of species in that particular area or the ecosystem. And for site A that we have seen, there are 7 species. And to know about the beta diversity, you have to count the unique species of both of them. So that means you discount the species that are present in both. So here we see between A and B, we have 8 different species. So the beta diversity is 8. And to know about the gamma diversity, you just count all the species in both of them. Okay, so for this, in this example, if we take just the site A and B, then it is 10. You can do it yourself between B and C, the beta diversity and the gamma diversity for A, B, and C. Okay. All right then. Now these are the levels. So what have we learned? We learned that there are 3 different levels of diversity. One is the genetic level, the species level, and the ecosystem level. And there are measurements, or to measure diversity, there are 3 different indices: alpha, beta, and gamma. Understood? Okay. All right then, let's go back to the board. Now, what are the attributes of, let's take another, hello. What are the attributes of this diversity? Now, this variation, these differences are more pronounced when you look at the latitudes. Oh my God, what's going on? Latitudinal, something going on. Sorry about that. Latitudinal gradient. There are 2 different attributes that we are going to learn today: latitudinal gradient and species richness. Okay, so let's look at the first one, latitudinal gradient. Okay, what are latitudes, people? Latitudes and longitudes we've learned, right? Looks like we are going into math, social studies. Today we are all over the place, huh? Okay, so if we take the globe, these that go across, these are latitudes, right? Remember, these are the poles. This is 0 degrees, right? So gradient means something that goes off from lower to higher or from higher to lower. So let's see what this latitudinal gradient is. So if this is the planet Earth, and this is the equator, we have taken some guys started doing research on how the birds, the variety of birds differ when we go from equator to the poles. So he had taken 3 different places. One is at the 0 degree latitude, which is today. I'm sorry, my pen is acting up today. At 0 degree, which comes around Colombia. 0 degree Colombia. He counted the number of variety of birds, and he came up with the number 400. Now, as you are going up, your latitudes also go up, right? And he took another place at 41 and at 71 degrees. This is south, right? This is north. Okay, so this, the 41 degrees is New York. And how many species did he find? He found 105 species of. These are birds, people. These are latitudes. These are. Okay. The research tells us that at 71, which comes around Greenland, he found only, God, what do I do with this pen? He found only, it's found only. So what do we see here? It means here from the equator, as we are going up from the tropics to the temperate zones, the variety of birds is decreasing, right? That means the diversity is going down, or the variety of the birds is going, not just birds, this is just an example, but usually the flora and the fauna go down as you go from equator to the poles. Okay, so this is the latitudinal gradient that we see when it comes to biodiversity. Okay, yes, okay. So the message here is the diversity is more in the tropics than in the temperate region. Okay, so let's try to see why that is so. What do you think? One of the reasons is now these temperate regions, they have been undisturbed for millions of years. Remember, these are the tropics, the middle where the equator goes through. These are the tropics. And then you find the temperate zones, and then you find the poles, right? So this region, this tropical region, has been undisturbed for millions of years. Or if you don't writing, you can hear me, right? The temperate and the poles, they've been bombarded with glaciers. So that's why species diversity is less in the temperate regions than in the tropical regions. Because the climate was undisturbed for millions of years, speciation took place. Speciation means formation of new species. New species have formed because there were no disturbances, either natural or man-made. Well, natural, because we're talking about millions of years here. So new species have come into existence, which is called speciation. We are looking at the Reasons for the diversity. More diversity in the tropics than in the temperate zones. Why? Because the first one is because these regions were undisturbed for millennia, for millions of years. The second reason is the climate is constant and predictable. They had a constant and predictable environment. So how does this translate to diversity? To do something about the pen. So here, because they have a constant climate, the animals behaved certain way. The behavior of the animals led to niche development or niche specialization. Because the climate was the same and a predictable environment. So they developed into different species. The third one is these tropics have an abundance of natural resources like solar energy. This we can actually relate to because we have, very hot summers and the rains too during the monsoons. So solar energy, the food, the water is in abundance. And so this is also a reason for diversity or variety of organisms. So to go over one more time, you know that as you go, we have seen that as you go from the equator to the from the tropics to the poles, that is the temperate zones or the poles, the diversity goes down, and it's the other way, right? If you're coming from poles towards the equator, then you know that the diversity increases. Okay, get it right. Whether you go from equator to up or from poles to down to the equator. Okay, so at 0 degree latitude, which is around in Colombia, you see 1400 species of birds. But as you go up towards New York and further up to Greenland, the number of species decreases, which is in New York you find only 105, and in Greenland you only see 66 different species of birds. That means your diversity goes down as you're growing up. Now that we have seen that the tropics have a wide variety of organisms than in the temperate zones, why is that so? There are three different reasons. What are they? One is because they have been undisturbed by natural calamities, because temperate zones were usually bombarded with the glaciers. Glaciations happened, and this did not occur in the tropical regions. And that led to speciation. That means new species have developed. That's why you have more diversity in the tropics. But that is only one of the reasons. Second reason is because we had a constant predictable environment. So there was niche specialization, which also led to diversity. And the third one is because we have an abundance of solar energy, food, water, and stuff. So new species develop. These are the three different reasons for the diversity that we see in the tropics rather than in the temperate zones. Okay. So so this is about this. This was we are talking about We are talking about latitudinal gradient. Now let's look at species richness. species richness. Wait, sorry about that. Species richness. Now, there was this scientist by name Alexander von Humboldt. According to him, he said that as you increase the area, the diversity also increases. The variety of species found also increases. But this is not a linear curve. This does not go up as you keep on increasing. At a certain level, this tapers off. So if we take a curve, if you start counting, there's a steep rise first, and then it levels off. Hey, happened here. And then it starts leveling off. It only goes up to a certain extent. Now, even if you increase the area, the number of species remains the same. That means you don't find no more new species even if you increase the area. Now this is called a rectangular hyperbola. Remember this. Huh? Rectangular hyperbola. This is not a straight line, right? So we usually don't understand if it is not a straight line. But remember, according to Alexander von Humboldt, as you increase the area where you want to count or where you want to see the diversity of the species, the diversity goes up. That means the variety of the species found in that area usually goes up, but only up to a certain point, and then it plateaus off. Okay, that means it doesn't go up anymore. So this is called a rectangular hyperbola. So the equation is S, which is species richness, oh my god, okay, S equals work now, S equals worse, S equals C times A to the power of Z. This is the rectangular hyperbola equation. Species richness denoted by S equals C, which is a constant, times area C times area to the power of Z. Now because this is a rectangular hyperbola, all the points here are clumped up and we can't actually learn anything. In order to get a straight line, we take log on both sides. So if we take logarithm on both sides, today's class seems like math. log x log s equals c plus d. Okay. So the actual equation is s equals c times d to the power of z, which is a rectangular hyperbola, which for our understanding, to make it clearer, if we take log on both sides, we get log s equals log c plus z log z log a. Okay, so the species richness s equals c is the y-intercept, z is the constant slope, and a is the area. Okay. So if we take this, now because this was a rectangular hyperbola, this, we've taken log and now we get a straight line. And that looks something like this. This is the slope. And the z would be, for this one, is 1. Okay. Usually the species diversity would be species richness or diversity would be between 0.1 to 0.2. That means it will be somewhere around here, 0.12. But if we take a large area, that means a continent, then if we take the frugivorous, there is a word called frugivorous, fruit eating birds and mammals together. For them, if we count a large enough area, this z, instead of being 0.1 to 0.2 for small ecosystems and for small areas, it would be 0.6 to 1.6. So somewhere around here, here. So that would be your species richness. All you need to understand in this is the diversity is more in the tropics than in the temperate regions, and there are 3 reasons for it: because they were undisturbed, a constant environment, and abundance of natural resources for the greater variety of species. And that is the latitudinal gradient. And for the species richness, as you increase the area, your species richness or the diversity goes up, but at certain level, at a certain point it levels off. And the graph that you get when you start off counting the species within an area, you will get a rectangular hyperbola. But if you take a log on both sides, you'll get a straight line. Usually the species richness is between 0.1 to 0.2 for smaller areas. But if you take a continent, then your diversity is definitely high, right? So that would come around 0.6 to 1.2. Okay. So the next is the importance of it. We'll just talk about the importance and then we'll stop. So why is this diversity important? Why do we have to learn so much about it? According to Tillman, another guy, Tilman, according to Tillman, if there's more biodiversity, then that ecosystem will be stable. biodiversity is more, then he says that that environment will be stable against natural and man-made calamities. Yes, that's what he said. He actually made the experiments that he did was he took a large enough area and he divided them into different plots and he did experimentation on it. So what he and then he grew stuff in here and he actually counted the varieties and also took the biomass. But so according to his experiments, if the diversity is more, then that environment would be stable. Not only that, he says that if the biodiversity is more, your productivity, whatever you are growing, would also be more. It will be more. It's increasing, and then you will have more or less the same productivity. So instead of getting less crop one year, more crop another year, if your plot has more diversity, then your biomass or the productivity would be the same. Okay, those are the two main reasons for us to actually understand this diversity and why is it important one, what kind of a role it plays um, in our day-to-day life. Okay. And is there time enough for us to look at rivet popper theory? Um, we'll talk about it in the next class. Okay, this will be it. All right. And don't forget to, this is just a regular class, but don't forget to look at the 3-minute videos that have been posted on YouTube, same channel. Okay. All right. Bye bye.